So one of the great mysteries of biology, remaining mysteries of biology, is how the one-dimensional code of the DNA molecule is translated into three-dimensional form. In the embryo, the genes are like management. They have the plans, they supply the materials, and they issue instructions, but it's really the cells and proteins that do the heavy-duty lifting of constructing an embryo. And so for about 30 years, we have dealt with this problem. So modeling, mathematical modeling plays a major role in pretty much everything we do. And as the statistician George Box said several decades ago, all models are wrong, but some are useful. This is particularly true when it comes to modeling of morphogenesis, which is the creation of biological form or shape. And two of the main issues that complicate this problem is that multiple mechanisms can produce essentially the same shape. And most of these problems include a large number of unknown parameters. So our main strategy then is to minimize first the number of free parameters, then determine the remaining unknown parameters using experimental data for normal development, and then with all parameters held fixed, test the model using data for perturbed development. But all of this is complicated further by evolution, as embryos have developed numerous ways to compensate for perturbations. So if we perturb the embryo, it's sometimes very hard to interpret the result of the experiment. So our long-term goal then has been to understand how the embryo creates organs. We mainly are concerned with organogenesis, to help researchers develop ways to prevent and correct congenital malformations. And so here I'm going to talk about the development of two organs, the heart and the eye. We also do a lot of work with brain development. So during development, the heart transforms from a single tube into a four-chambered pump. The first stage of this process is called looping, as a straight tube deforms into a curved tube. This brings the future atria, shown here in purple, above the future ventricles down here. Then septation divides the tube into four chambers. So here I'm going to talk about first how the heart forms in the first place, and then how it loops. In humans, looping takes place during the 4th week of development, just after the heart begins to beat. In our experimental model for this process, the chick embryo, whose heart develops very similarly to that of humans, in the chick, looping takes place during day 3 of a 21-day incubation period. Now, this is an image of an embryo, a chick embryo, after 24 hours of incubation. It's going to be cultured for another 24 hours. There's not yet a heart or not much of anything, but this darkened region up here is a membrane that's folding on both sides toward the embryonic midline, the middle of the embryo, where they're going to fuse and form a heart. So I'll run this over. This is about 24 hours. So this is the heart tube being formed, elongating, and then looping off toward the right side of the embryo. Then just a day later, the heart is beating with much more force, and blood is being pumped. It comes in through the bottom, goes around the loop, and out through the top to the aorta, and it pumps blood to the rapidly growing embryo. Just for your coordinates, this is the head, and this thing is the eye of the embryo. So the heart first forms from a membrane called the splenic pleura, which consists of a layer of mesoderm attached to a layer of endoderm. The primitive heart fields are in these dark red regions on both sides of the mesoderm, and this is what the heart fields look like looking from above. For a long time, it was commonly thought that these membranes fold directly toward the midline, both sides fold directly toward the midline, where they fuse and separate, with the mesoderm in red expanding to create the heart tube, and the endoderm at the same time in blue forms the primitive foregut. And notice how the heart comes in contact with the remaining splenic pleura outside, that's left outside the heart. But about 15 years ago, Margaret Kirby's group found that the heart fields don't fold like this. Instead, they fold diagonally toward the midline, and this brings the outer portions of the heart fields to create the lower part of the heart tube, and the inner portions in red create the upper part of the heart tube. So let me see if I can, I'm going to stop sharing for a second so that I can illustrate this. Actually, can you see me?
Yeah, no problem.
Okay, let me see. Maybe I can just do it this way. So this is a piece of paper with the red in the middle and the blue on the two outer and the two sides. So for many decades, it was thought that the folding just occurred like this, or just rolled up, and so you have blue on blue. You have a region of red in between two regions of blue. Okay, but Kirby found that this paper folds actually like this diagonally, and so you end up with, see that, with red on top and blue on the bottom. Diagonal folding. And so our objective then, oh, and because the endoderm is attached to the mesoderm, both layers must fold the same way. And so our objective here was to determine the forces that create this kind of folding. So first we went to the lab and did some experiments with chick embryos. This is a control embryo on top that's cultured from stage 5, when there's not yet any folding, there's not much here at all, for 15 hours, to stage 12, which is the end of, toward the end of the first phase of looping. And then you get a heart tube like this that's looped to the right side. Notice how this darkened region, again, is the folding part of the membrane. As the heart tube elongates, it's indicated by this dissension of this arch-shaped on the bottom anterior intestinal portal, which is the opening to the foregut, and so it goes down as the heart tube elongates. And it turns out that in St. Louis here we have a very large replica of the anterior intestinal portal. Now this embryo was exposed to the mitotic inhibitor. So in this case, we're studying the effects of growth. So we exposed the embryo to the mitotic inhibitor, acetylcholine, for 5 hours. Then the drug was washed out and incubation continued up to the 30-hour period. So during the exposure to the drug, notice how little or no folding had occurred. Then after the drug was washed out, the folding started up and continued relatively normally to form a heart tube, even though the heart tube ended up with a rather abnormal shape. But what this experiment indicates that the initial folding requires cell proliferation or cell division. Okay, so growth is one of the main processes that embryos use to create force. Another process is contraction. There are really two main processes, growth and contraction. So to study the effects of contraction, we exposed embryos to the myosin inhibitor blebbistatin. So this embryo was exposed to blebbistatin for 7.5 hours. Then the drug was washed out. Incubation continued for another 7.5 hours. So as you can see, the drug had little effect on the folding, the initial folding. But what it did have an effect on was the rate of folding. And so what we did to estimate the rate of folding or the rate of elongation of the heart tube is measure the distance from the top of the head to the top of the anterior intestinal portal. And here in the blue it's a control embryo plotted as a function of time. So this is the relative elongation of the heart as a function of time. And here it is exposed to blebbistatin for 7.5 hours. You see the rate decreases to almost a stop. Then after it was washed out, the rate picks up again and becomes almost normal. So what this experiment seems to indicate is that elongation of the foregut and heart tube requires actomyosin contraction. So putting these results together, we proposed a hypothesis for heart formation, or for the folding that creates the heart. First, it's important to realize that if we have a bilayered membrane like the sphygmopleura, pleura, and if the top layer grows, it will force the membrane to bend with the growing layer on the outer curvature. And so to get anisotropic, orthotropic, or diagonal folding, we assume that the growth is orthotropic, with the bottom layer, original bottom layer, growing at a faster rate than the top layer, this would cause a diagonal folding toward the midline. And after fusion, we speculated that contraction along the anterior intestinal portal, the edge of it, generates a tension that pulls the heart fields downward and causes forcing them to elongate, with growth contributing to the elongation. So as a test of the plausibility of this idea, we created a relatively simple finite element model for the splenic pleura, consisting of a layer of endoderm attached to a layer of mesoderm. This is originally a rectangular flat plate, and we only needed to model half the embryo because of symmetry. So if we look at both layers separately, this is the mesoderm. The heart fields are located in this upper corner of the mesoderm. And this is the y-axis is the axis of symmetry. And the border of the anterior intestinal portal, the future border, will become this green stripe along this edge of the endoderm. We also assume that the bottom edges are on rollers and the other two edges are free. Now we simulated growth and contraction using the theory of Rodriguez et al., whereby the total deformation gradient tensor is decomposed into an elastic deformation gradient tensor F star and a growth tensor G. The growth tensor is something like a thermal or a plastic strain. It's analogous to those. But the stress then depends only on the elastic deformation. And so the growth can be either specified as a function of time and place or determined from a growth law, where maybe it depends on stress or something like that. In all the models I'm going to show today, growth will be specified everywhere. And then contraction can be simulated as negative growth with also an increase in regional stiffness, much like a muscle contracts, it becomes stiffer. Okay, so this is the model in the initial configuration, and I'm going to run this model. Okay, so first you see the diagonal folding. When this free edge hits the midline, this edge is transformed into a roller support, and then contraction is going to be turned on along the green stripe here. So now the contraction is turned on, it pulls everything downward, and then the layers separate, and pressure inflates the heart tube, which is the red. This is a cross section of the model at one location. So the initial folding toward the midline. And when it hits the midline, the layers are going to separate, and the heart tube expands and comes in contact with the splenic membrane outside. This slide shows a comparison between the geometry of the heart of the model with that of an actual embryo. So first you can see that the model qualitatively captures the tapered shape of the heart, and it also includes this continued folding region down here. Are these two veins, ophalomesenteric veins, that continue to fold and form the bottom part of the heart tube. These are 4 cross sections located as indicated here along the heart tube and in the veins, and these are the same, apparently qualitatively the same cross sections in the model. So if the heart here is stained in dark, you can see the heart tube in each cross section, the dark region, and compare that to the red heart tube here, and you can see that it has a similar shape in each cross section. The shape of the foregut also has a similar shape to what is seen in vivo. This is the foregut here, even though it's not quite as flat. So this seems to at least give tentative support to our hypothesis. Okay, now I'm going to turn to the problem of looping. Once the heart has formed this tube, now it loops, and this is a very important process. This is the first sign of left-right asymmetry, major sign of left-right asymmetry in the developing embryo. If looping goes wrong, the heart typically will not develop correctly. So in this experiment, I'm going to focus only on the first phase of looping, which is called C looping, as a straight heart tube deforms into a C-shaped tube with outer curvature almost always directed toward the right side of the embryo. In this experiment, we put tissue labels along the ventral surface in the straight heart tube. Toward the end of C looping, they move toward the outer curvature. So this indicates that looping is not just a bending of the heart tube to the right side, but it's a combination of a ventral bending, outward bending, and a rightward torsion. So if I can show, here's my heart with some labels on it. So it bends outward and then rotates or twists to the right side like this to move the labels to the outer curvature. And this is what a cross section looks like during this time. The wall of the heart tube is composed of 3 layers. There's an outer layer of developing heart muscle called myocardium. There's a middle layer of extracellular matrix called cardiac jelly. And then there's a 1 cell thick endocardium that lines the lumen of the heart tube where the blood is. And so again, I just want to remind you that the heart begins to beat during this time, but there is not yet any significant blood flow, so the embryo can remain alive even without the blood flow during these stages. Okay, so the bending and torsion are coupled to some extent, but it's useful to consider them separately. So during the last 100 years, several hypotheses have been proposed for bending. These include a constrained growth, whereby the heart simply outgrows its allotted space, and that forces the heart tube to buckle and bend. Differential growth by cell division, which is also called hyperplasia, where the cells on the outer curvature grow faster than those on the inner curvature, forcing it to bend. Active changes in cell shape. It's known that the cells on the outer curvature become longer longitudinally, those on the inner curvature become shorter, and this would force it to bend if this was an active process. Cardiac jelly pressure of dorsal constraint. This was proposed by Frank Manisek, because he noticed that the heart expands as the cardiac jelly inflates during looping, and it's also attached to the foregut initially as it starts to bend. So he said, well, it'd be like blowing up a balloon with a piece of tape on one side. And this is a piece of tape, this is a picture from one of his papers showing a balloon that he blew up with a piece of tape along the inner. And then finally, there's differential contraction, where the cells maybe along the inner curvature contract longitudinally, forcing it to bend. But over the last 100 years, experiments have essentially ruled out all of these possibilities with the possible exception of active changes in cell shape. And when we first started to study this problem, we studied this problem starting around the year 2000, maybe a little bit before that. We thought that this was what was happening. We thought we had evidence showing that this was what was happening. But then along came this paper about 15 years ago by Soufan et al., where they found that the heart cells change size during looping, with the cells along myocardial cells along the outer curvature becoming about 2 to 3 times larger than those along the inner curvature. These results came as something of a surprise, because prior to this time, it was commonly thought that the heart grows exclusively, almost exclusively, by cell division before growth and by increase in cell size or hypertrophy after growth. And this was why some people had before ruled out differential growth, as I mentioned in the previous slide, because they could not find significant patterns of cell division over the heart tube during looping. But these results showing increases in cell size led us to reconsider the possibility that differential growth is a cause of bending. So bending is more conveniently studied by considering isolated hearts that are cultured, because they bend in culture without the complicating effects of torsion. And notice how this darkened region is the dorsal mesocardium, the attachment to the foregut, which actually ruptures during looping. And always, in all cases, this becomes along the inner curvature of the bent heart. So this is a culture over a period of 12 to 24 hours. So to test this idea, Yunfei Shi, one of my students a few years ago, created a relatively simple finite-element model for the isolated heart tube, consisting of a circular cylindrical shell with the inner core of cardiac jelly. First, he specified isotropic growth in the jelly that causes the jelly to inflate. and puts the myocardium under tension in red here. And then he specified growth around the circumference in a patterns that was consistent with what was measured by Soufan et al. And this causes the heart tube to bend with the original dorsal side located along the inner curvature as it should. And it also has a right amount of bending. To test the model further, because as I said, different mechanisms can produce the same shape. And so it's important to use other tests to examine these problems. First, he compared measurements of strain to those predicted by the model. And he measured, for example, longitudinal strains along the outer curvature and inner curvature by tracking the motions of tissue labels over a period of 24 hours, during which time the heart loops in culture or bends in culture. And these are stretch ratios plotted in the radial direction, the circumferential direction, and longitudinal direction along the inner curvature and outer curvature and inner curvature. The radial strain was estimated by measuring the thickness changes of the myocardium, circumferential strain by changes in its circumference. And as you can see, except for the radial strain, which it predicts, the model does a very good job of predicting the magnitudes of these other strain components. Now, to estimate stress, to characterize stress, that's a more difficult problem, but we do this by making small cuts in the myocardium and measuring the aspect ratio of the resulting wound after a few minutes. So, for example, in this heart, there was a small cut made in the longitudinal direction, in the circumferential direction, and it opened longitudinally to indicate the magnitude of the longitudinal tension. So you do this at the beginning of the experiment in one heart, and because this would change the development, we have to do other hearts and cut it at the end of the experiment. And so this down here are plots of the change in aspect ratio during this bending in the longitudinal and circumferential directions. For this, these are indications of the direction of the stress, outer curvature and inner curvature. And along at the outer curvature, the model is very good at predicting the change in aspect ratio. It underpredicts what happens at the inner curvature, but more importantly, it predicts the right trends. The model predicts that the stress decreases in both directions, and in both locations. And these agreement and trends seem to support the differential growth hypothesis. Now, if I turn to the torsion problem, so we found that, again, that bending is probably driven by loads intrinsic to the heart tube, but we've also found that torsion is driven by mainly by external loads applied to the heart. Some of these loads are applied by these two ophthalmomesenteric veins as they push against the bottom part of the heart tube. And this is a cross-section of the embryo showing the heart tube. It's attached again to the foregut by this dorsal mesocardium initially, and that constrains the motion of the heart. And then the splenic pleura is under tension and presses against the ventral surface of the heart tube. So to examine the effects of these external loads, we cultured hearts. This is cultured the whole embryo. First, we put labels along the left and right edges of the heart tube, cultured the embryo for 6 hours, and the heart tube, the labels move away from the two edges. The green moves to the top surface of the tube. The red, this side moves to become the red labels, move actually on the bottom surface. You can see it through the heart. And this motion away from the edges indicates the amount of rotation of the heart tube. Then the splenic pleura is removed from over the heart, and the labels move back toward the two edges. And then the two veins were cut off, and the labels return to their original locations along the left and right edges. So this simple experiment indicates that the splenic pleura and these two veins drive early torsion. So from this experiment, we proposed this hypothesis for torsion. There's a schematic showing a ventral view of the heart tube up here, a cross-sectional view down here, and it shows the splenic pleura that pushes against the ventral surface. So we speculated, we know that the two veins grow at different rates, with the left vein growing at a faster rate than the right vein. So we speculate that it pushes with more force against the bottom of the heart tube. This gives the heart tube a little bit of a jog toward its right side, and then the splenic pleura pushes the heart backwards, causing it to rotate about its attachment to the foregut. And so if this was the case, then we would predict that if we could lower the tension, the force in the left vein, below that in the right vein, we should be able to cause the heart to loop to the left side instead of the right side. So in this experiment, this again, culture in the embryo, a portion of the left vein was dissected away. The splenic pleura is still on top. And then the heart was cultured for 6 hours and it looped to the left side. And this happened in most cases, the majority of cases looped to the left when this experiment was done, consistent with our hypothesis. And this in vivo, in normal, under in vivo or in vitro conditions, you find that the heart rarely loops to the left. It almost, more than 99% of the time, under normal conditions, it will loop to the right. So this was very significant, consistent with our hypothesis. Now putting everything together, Yunfei created a three-dimensional finite element model for the heart tube, including the veins. The initial configuration, initial geometry was reconstructed from serial cross sections like the one shown down here. The heart and veins were placed between two membranes to represent the splenic pleura in front and the foregut behind. If you look at a side view of the model, the back side was put on roller supports at the top and bottom. This part between them is free as the dorsal mesocardium ruptures during looping. This is a typical cross section of the model. Look at its shape, compared to one from this image. It looks very similar. It consists of, so we include the cardiac jelly and the myocardium. And this is a longitudinal section of the model showing the distribution of the jelly and the myocardium. And it also has a lumen. You can see the lumen in here. Okay, so this is the model. So again, we specified a distribution of growth in this green region, the heart tube, consistent with Sufan's measurements, Sufan et al.'s measurements. We specified growth in the top parts of the two veins and contraction along the anterior intestinal portal, the bottom parts of the veins. To visualize torsion, these labels are fixed to the model. And I'm going to run this model. And it starts off a little slowly, but then it starts to bend outward and rotate toward the right side. And if we compare the final shape of the model to a typical heart, you see that it has a very similar shape. Notice the shape of this vein here, and the labels are in the correct locations. So this gives pretty much confidence that at least we're on the right track with this hypothesis. Okay, as a further test of the model, we again compared model and experimental results, this case for strains, and these are longitudinal strains that are average over 7 different regions of the heart and the veins. So in each case, up here, you can see that the general trend predicted by the model is correct for each location, with the possible exception of the bottom parts of the vein. So it's not perfect everywhere, but it's at least reasonable. And also I should mention that the comparison, qualitative comparison of strains, of stresses by cutting is also reasonable. But now it's really important to take a look at how the model can predict what the behavior under perturbed conditions. So first we did an acute perturbation over a period of just several minutes. So in this experiment, labels were placed along the left and right edges of the heart tube. Same was done in the model. Culture the heart to the end of C looping. And again, so the labels, as I showed before, they rotate away from the edges. The same happens in the model, to similar locations. Next, the splenic pleura was cut off from over the heart. It was removed from over the model. And as you can see, the labels move back toward the two edges as it does in the embryo. And then in this experiment, then the top part of the heart was severed from the embryo. The heart tilted like this, And the same thing when you simulated this experiment happened in the embryo. And then we tested the predictive capability of the model over a period of during the period of C looping, as during morphogenesis. So in this experiment, labels were put on the midline, again, to visualize rotation. Then either the left vein, the right vein, or both veins were cut, and the heart was cultured for 12 hours to see what happened. And the labels moved to indicate the rotation. We simulated the experience with the model. It's important to realize that in all of these cases, when we test the model, the parameters in the model are kept fixed. The only difference here is that either the left vein or the right vein or both veins were removed, and the model was then run. And then we compared the final shape to what we saw experimentally. So in this case, you can see that if the left vein was cut off, the heart looped and rotated toward its left side, abnormally towards left side, just as it would happen in the experiment. If the right vein was removed, it rotated rightward, same in the experiment. Notice the abnormal shape of this vein is captured somewhat by the model, the remaining vein. And if we remove both veins, the model rotated rightward as in the experiment. So we're pretty confident in that because of these results that we have a reasonably good handle on the C looping process, and it took us 15 years to get to this point. Okay, so like the heart, the brain begins as a simple tube that soon divides into to these subdivisions called the forebrain, the midbrain, and the hindbrain. The eyes initially are a pair of optic vesicles, like spherical optic vesicles that grow outward from the two sides of the forebrain, and they come in contact with this overlying membrane in green called surface ectoderm. Here shows a close-up of what happens with the eye. This is the optic vesicle and the surface ectoderm. When they in the region of contact, both layers then thicken to create what are called placos. And then they invaginate or bend inwards together with the optic vesicle in yellow forming what's called the optic cup, which later on becomes the retina, and the surface ectoderm forming the lens vesicle, primitive lens. This is a cross section of a chick embryo showing the lens included inside the optic cup here. And this is what the adult human eye looks like with the lens focusing light onto the retina and back, which just becomes a very thin membrane at the end of development. So I'm going to talk about the formation of the optic cup, and then if there's time, I'll talk about the lens. So our main problem here is what causes the optic cup, the optic vesicle to invaginate. The question is it regional contraction? As many invaginations are caused by contraction. And so the wall of the eye is a single layer of cells, epithelial cells, that have an apical side and a basal side. On the apical side are located actomyosin fibers that can contract. And typically in many invaginations, these fibers contract, causing the layer to bend with the contractile fibers along the inner curvature. And so we look for these fibers in the eye. This is an embryo stained for actin. And this is a blow-up of the eye. The actin is the bright lines here. This is the lens. Notice how the actin is along the inner curvature of the lens, and which is the right side needed to cause it to invaginate. And experiments have shown that the lens does indeed invaginate because of this kind of contraction. But it's on the outer curvature of the optic cup, which is the wrong side to cause it to invaginate. And because of this, we can rule out contraction for this process, for the optic cup, for the optic vesicle. And this has led some investigators to speculate that the lens, when it bends inward, simply pushes the optic vesicle inward, causing it to invaginate. But experiments by Jeanette Heyer's group has seemed to contradict this idea. And so we repeated these experiments to take a closer look at this problem. So Alina here, Alina Otain, removed the surface ectoderm at different stages of invagination and then cultured the embryo overnight. So if the surface ectoderm was removed before any placos had formed, after an overnight culture, there's no invagination. But if it's removed after invagination had begun the next day, a relatively normal-looking optic cup had formed even without a lens. The lens would be forming from this layer was left up here. Okay, and so this shows that the lens invagination is not needed for optic cup formation, but the surface ectoderm is needed until the optic vesicle begins to invaginate, and then it's no longer needed. So then from this idea, these data we proposed what's called the matrix constraint hypothesis. First, it's important to realize that the two layers, the optic vesicle and surface ectoderm, when they come in contact in the region of contact, they're attached by a relatively thin extracellular matrix that's secreted by both layers, but mainly by the surface ectoderm. And so we speculated that this invagination of the optic vesicle is caused by the growth of the cells constrained by a relatively stiff matrix. So then if you have cells that are dividing, tend to making grow longer, the matrix does not grow, and it forces the layer to bend with the matrix along the inner curvature, as happens here for the optic vesicle. And so if the surface ectoderm is removed too early, then we thought, well, maybe not enough matrix has accumulated to cause it to invaginate after the surface ectoderm was removed. But if it was removed later, after invaginated started, then there's already enough matrix present to cause the vesicle to invaginate, even without the lens. To test the plausibility of this idea, Alina developed a finite-element model for the optic vesicle. In the initial configuration, it's a spherical shell now, composed of mostly of a layer of cells. And in the region of invagination, there's a thin matrix layer along the outer surface, just in this region. And the material properties of the matrix and the cells were determined by micro indentation. And it showed that the matrix has a modulus of about a factor of 10 times larger than that of the cells. Growth was specified equally in the circumferential and the meridional directions in the shell, with a faster rate of growth in the invaginating region. When I turn this on, you can see how the curvature reverses and continues to invaginate a long way. And in the final shape, looks much like the form of an optic cup. For further validation of the model, she measured the relative invagination depth as a function of time, the thickness at the center of the optic vesicle as a function of time, and the curvature at the center as a function of time. And in all these cases, the model in blue agrees relatively well with the experimental data. Now, you might notice this little blip here in the thickness. This is caused by a local, as the curvature is reversed, there's a local a snap-through type of buckling that occurs, and we could not discern as to whether or not these dots are showing this kind of buckling. It's not accurate enough to show this, but that's what the model shows. Anyway, it seems to at least help support the view that this differential growth, constrained growth, is what causes optic vesicle to invaginate. To test the model, these experiments were conducted. First, the surface ectoderm was removed, and then the embryo was exposed to collagenase to remove the collagen and essentially remove the matrix. So essentially, we're looking at what happens when the matrix is removed. If this is done after an early part of invagination, notice how the curvature reverses as the optic vesicle pops back out from where it was. But if it's removed during mid-invagination, it remains invaginated. And in fact, what we found in general is that the later the embryo, the later the surface ectoderm was removed, the fewer optic vesicles, optic cups that pop back out to reverse curvature. And so this experiment suggests that the matrix is needed to maintain optic vesicle invagination during the early, but not the later stages. So then we simulate these experiments using the model. So the model was run to a certain stage of invagination, then the computation was stopped, and gradually the properties of the matrix, the growth and material properties of the matrix were gradually changed until they matched that of the cells below. This effectively removes the matrix, and when this was done during early invagination, you see how it popped back out. But if it was done later on, it remained invaginated, just like in the experiments. And what happens here is that the growth of the cells here has gotten so big that this can no longer fit through the hole that was created when it initially invaginated. So it just can't go back out. And then more recently, Hadi Hosseini developed a three-dimensional model for this problem. So this is the initial configuration. This is the optic vesicle. This is the surface ectoderm, just a shell on the outside. This is a time lapse of the model. I'm just going to show just the optic vesicle. So it's going to grow outward. It's going to come in contact with the surface ectoderm, which is going to force it to bend downward. And the growth was specified similar to what was done, or consistent with what was done in the previous axisymmetric model. So you can see it bends downward and starts to invaginate. And now if we compare the final shape against, we flip it up, compare the final shape against the schematic of the optic cup, you see it has a very similar shape, and including, because of the three-dimensional properties, this groove here is consistent with what's seen, this groove called the optic fissure, which later on becomes a conduit for blood vessels. So I can stop here. It would take me 5 more minutes to go to lens development, but if you want me to stop, I can stop at this point.
No, please, you can finish up.
Okay. So I'm going to talk just a few minutes about the lens. So as I said before, the lens is known to invaginate because of actomyosin contraction along its apical surface. So we didn't want to look at this problem any further. But one question that was remain pretty much undecided was, how does the lens vesicle close? This problem is not as simple as may at first appear. If the lens was a portion of a cylinder, then contraction along the inner curvature could easily close it, like bending up a piece of paper is so easy to do, rolling up a piece of paper. But the lens is actually more like a sphere with a hole inside that needs to be closed. If we look at a top view, as we pull in all sides of the hole to make it close, we have to shrink the circumference down from a finite size to essentially 0. And this causes a significant compression outside of this region, which resists closure. And in fact, it makes it very difficult for it to close, to resisting the large compressive stresses. And this is similar in the embryo to what happens when you punch a hole in an epithelium. What happens is that the embryo is developed, and so to close the wound, the embryo has developed a nice way to generate more force to close these wounds. So if you punch a hole in epithelium, shown here, what happens within a matter of just seconds, a ring of actin forms around the hole and starts to contract with myosin, and it closes the wound within a period of several minutes, and it closes completely without leaving a scar. And so the wound heals completely without leaving a scar. And so we look for such a ring around the opening to appear to the in the lens, but we could not find one. And so it must be something else. So there's no obvious contractile ring surrounding the hole in the left vesicle, in the lens vesicle in this case. Okay, so we noticed that for several decades, it had been known that a region of programmed cell death called apoptosis develops around the opening to the lens as it closes. but nobody knew the reason for this. And so we speculated that what this does is simply remove cells around the hole, which would shorten the circumference and forcing the lens to close. And we had tentative support for this idea because when we exposed embryos to apoptotic inhibitors, most of the lens stayed open. So to test the physical plausibility of this idea, Alina then developed a finite element model for the forming lens. So in this case, the initial configuration was a spherical cap obtained by rotating this section about the axis of symmetry here. On the top, there's a thin layer of contractile fibers. On the back side, there's a matrix similar to what's seen in vivo. And the future region of apoptosis will be out here on this end here. So first, the contractile fibers were contracted, were activated. That forced the curvature to reverse, and the lens started to close, but only to a point. It could not overcome the compression alone that developed in the lens. And then apoptosis was simulated as a negative circumferential growth around the hole, which is here, and this caused the lens to close. And this is a three-dimensional representation of the final shape of the model. And this is a comparison of the model in red, the cross-sectional shape, to the embryo, which are these bright colors, and it had a very similar shape to the actual lens. And then Alina went on and measured the normalized thickness of the center of the lens as a function of time, as well as the arc length on the basal side and the apical side as functions of time, and also this gap distance as a function of time. And it seemed to predict reasonably well the behavior in all these cases. So anyway, in conclusion, our models are definitely wrong, but we hope that they may be useful. And so here, most of the people that are involved in this, in the three studies I showed, are shown here. And these are the other members of my team since I moved to St. Louis in 1997. These are graduate students who worked on morphogenesis in particular, on these three problems also, and some of my collaborators. And thank you very much. I can take any questions.